question is, do they do different things? So we've got these two versions of MDMA, S and R, the left-handed version and the right-handed version, but we'll just call them S MDMA and R MDMA. Many drugs have these two versions. This is very common. Ketamine is like this. LSD you mentioned has four different versions because it's an even more complex structure. So you've got two versions of MDMA. The question is, do they do different things? Do they have different behavioral effects in animals? Do they feel different when people take them? Do they have different effects on plasticity, and so on and so forth? There was some hints in the literature that perhaps S and R MDMA had different effects in animals, but the results were a little ambiguous or conflicting. But a priori, like without knowing anything, is it common for different isomers to have different effects? Like would we expect them to be pretty close to identical in their effects, or would we expect them to have some kind of difference in their effects?
Usually they behave differently. I mean, both molecularly in tissue culture, in cells, and also in animal models. They usually induce different effects behaviorally and in terms of signaling, cell signaling.
And so the other piece of background here that I want to discuss before going into this paper is, okay, so on the one hand, we have got the fact that drugs can have isomers or different versions of each other. There's SMDMA and RMDMA. On the other hand is the concept of sex-specific drug effects. So males and females may or may not respond the same way to the same drug or the same isomer of one drug. Sure. Can you just talk about that a little bit? How common is it for there to be sex-specific effects for drugs in terms of their behavioral effects and their pharmacological effects? Is it more often than not true that males and females respond differently, or at least not quite identically?
In the past, that's a very good question. And in the past, let's say 5 years or so, we've had many findings on in mice showing very, I think, interesting and intriguing sex differences between male and female mice. I mean, there was something about the story. So before that, most of the labs, we used only males because for reasons that basically, I don't know, but we used males. And one of the students, PhD students, Elena Yaster, so she told me that why do we don't test to compare experiment design to test to compare the effects, not MDMA. Back then was psilocybin, psychedelic, and DOI on behavior in male and female mice. So we tested that. It was a cohort. They were matched of male and females, the same age, all little mates, and intriguingly, the females in the behavior that we tested, one of the behaviors, head twitch, we can talk about this further, but with that behavior, with the same dose of psilocybin and also DOI, another psychedelic, the females responded more.
Responded more.
Yes, responded more.
And that would imply, so the head twitch response in a mouse is used as a proxy for the hallucinogenic or psychedelic effects. That would imply that in your guys' hands, psilocybin and DOI were causing a stronger psychedelic effect in females at the same dose compared to males.
But, for example, again, that is for the trip, if I can say that. For the again, it's a model. We know that mice have many limitations, but that is, as you said, the behavior, the proxy that we use. We know that there's a nice correlation between the head twitch and the psychedelic effects in humans. So, and we also know that's important that the head twitch, it is principally serotonin dependent. So we know that those effects are serotonin dependent. But again, that was when we were testing the psychedelic or the hallucinogenic effects of these drugs. But when we were testing the therapeutic effects, for example, measuring reduction in models of opioid addiction, or measuring the effects of psychedelics or the effects of MDMA on dendritic spines, in most of the phenotypes, we don't see these effects in females at the same doses. So, I mean, again, those are the data. I think that it is very clear that there's a difference, at least in mice,